The German neurobiologist Frank Bradke has been awarded the Remedios Caro Almela Prize 2023 for his excellent career in developmental neurobiology. He is visiting the Institute for Neuroscience, a joint center of the Miguel Hernández University of Elche and the Spanish National Research Council, to give the Caro Almela lecture, in which he has presented his scientific achievement, and to attend the award ceremony. We took the opportunity to talk with him about his work. Welcome, Professor Bradke. Congratulations. You have just received the Remedios Caro Almela 2023 prize for research in developmental neurobiology. How did you feel when you received the news, and what does this recognition mean to you?
So thank you very much, first of all, for your kind words. And I actually got the news on the airport in Pisa. So we were just on the way flying out, and I checked last time my email, and I said, oh my God, so I got the Remedios Caro Almela Prize, and I was very excited. And my family was with me, and so we were already in the waiting line to board the airplane. And my wife, she found somewhere to get some Prosecco, and so we were still in the line, also we were cheering also on the prize. Yeah, so I was very happy, very excited, very overwhelmed.
And what does this recognition mean to you?
So I think for me this is a very special recognition, and actually I really feel humbled by the selection committee. So many of the former laureates, and they are, for me, personal role models. They are my scientific heroes, and for me, it's a very special to belong now to this group of people.
I have to ask: do we fully understand how neurons grow during development and how communicate with each other?
I think there's lots of space for students and postdocs to explore. I think it's a very young and a very vivid field where a lot of exciting discoveries await to be taken. And I think we do understand now a little bit more. And I think also our lab was also participating in this. But I think what I really find exciting is there's so much more that we can learn that is very good.
You led the group Action, Grow and Regeneration at the German Center for Neurodegenerative Diseases. And your discoveries about the nervous system has allowed you to do pioneer work in the field of action and regeneration in pathological states. How is it possible to regenerate nervous connection when they are damaged?
Yeah, that is something which we are still asking every day, and I think if you look in the adult nervous system, it is very difficult in the mammalian nervous system to get any regeneration. But what is very interesting is that in principle we know it should be possible, because during early development, so when we are embryos, the axons they grow really quickly to wire the brain, and so you have really these two sides of the same medal, that in the adult axons fail to grow, fail to regenerate, but during development in the young, in the embryo, you have this growth, and this is really what makes us excited. So we want to understand how neurons grow their axons, so how do they basically grow this biological wire, and if we understand this well, then we should also be able to reactivate these mechanisms under more pathological conditions to induce regeneration.
Your lab has a special interest in the skeleton of the cell, the cytoskeleton, yes? Yeah. And your team found out that microtubules play a key role in the growth of actions. Can you explain to us what are these microtubules and what is their function?
Yeah, so imagine, for example, if you build a tent, and so you need the poles to basically give structure to the tent. And these are in principle the microtubules. Yeah, so they are the pillars and that hold basically the tent intact. And on the other side, you have often at a tent that you have this constraint, right? And this is, and you can imagine that this sort of, it's like a net, and this net, through the tension and the interaction with the poles, so that they give form to the tent. And in a way, you can imagine that cells, they have their morphology in very similar ways. Yeah? So they have basically these poles, the microtubules, and they have the tent, which is often the actin cytoskeleton, and together they give forms. And the neurons are very special because they have many microtubules really bundled, and by this way they are able to make very long processes in the nerve cells, and they have these long axons which basically can cover the length of our own body.
What happened when these microtubules fail or damage or?
Yeah, so imagine now if you would have a tent and the poles would be basically destroyed, right? And this is happening after an injury in the spinal cord. There, the microtubules, they basically start to disassemble. And with this, you also have then a degeneration of the axon. And it's really, if you also think about, you would have these poles in a tent, and now the poles would start to disassemble, then the whole tent would start to collapse. Exactly. Yeah.
You led a young and diverse team at your lab, and I would like to end this interview by asking for advice for young researchers who are starting their careers in basic research. What do you tell them?
Well, so I have to admit that I also learn a lot from the young researchers, and so I'm often, I feel not super comfortable to give advice, and but I really like also to listen a lot to what they have to say. However, if I could give like one small advice, then it may be that one also thinks about the question that really interests you. And sometimes when you ask the scientific question, it may be also helpful to think, well, is it something, first of all, it should excite you? And second, you should also ask, are other people are asking the same thing? And so what I usually do is something, what I call the beer on the beach criteria. And that means, would it make a difference to work in the lab or to go to the beach and have a beer? Because in some instances you would find out that the knowledge will be produced anyway by other labs, right? And so, and then if you feel that the project would be done by many other labs anyway, maybe then it's not the most interesting thing that you would like to follow, but you rather would like to see what is really what you find personally very interesting, and to basically really do your science in this way. Does it make sense?
Yes.
Yeah.
Well, thank you very much for your time. Enjoy this recognition. We will follow the next steps that your research takes in the future.
Oh, thank you very much, Elena. Yeah, thank you.
Dankeschön.
Gracias.